Hello everyone, the title of my presentation is Effects of Acaricide Perizin Treatment on Brain and Midgut Gene Expressions of Honeybees. Perizin is an acaricide used in honeybees to control Varroa destructor, a highly virulent parasitic mite which is one of the main reasons for honeybee colony losses. It contains 3.2% comephos as an active ingredient. Comephos is an organophosphate compound which is a powerful irreversible acetylcholinesterase inhibitor. Negative effects of perizin and its active ingredient comephos were reported previously, especially increased mortality and impairments in behaviors such as food transfer, foraging, and learning abilities. We previously treated some native Turkish honeybee species with sublethal doses of perizin and subjected them to some behavioral assays. And in Syrian honeybee subspecies, which are endemic to southeastern Anatolia, we found increased locomotor activity, decreased olfactory conditioning, and decreased electric shock, or in other words, pain and threat avoidance rates. However, in Caucasian honeybee, which is endemic to northeastern Anatolia, we found no significant difference in treatment and control groups. And to understand the reason of this difference between these subspecies, we carried out a gene expression study by quantitative real-time PCR in brain and midgut samples of these two subspecies. We determined 9 candidate genes: nicotinic acetylcholine receptor subunit alpha-10, muscarinic acetylcholine receptor, and vesicular acetylcholine transporter, all of which belong to the excitatory cholinergic system, and also 3 GABA receptor subunits, which are involved in the GABA neurotransmitter system, which is one of the main inhibitory neurotransmitter systems in central nervous system, and also 3 cytochrome P450 genes, which are found to be specifically metabolize the active ingredient comephos in honeybees. Our brain gene expression results showed no significant difference between control and treatment groups, except a near significant decrease in treatment group of Caucasica in GABA type B sub1 receptor subunit 1 loci. To make a better comparison of subspecies, we normalized treatment data of each subspecies to their respective control means. Comparison of this data also gave no significant differences, unfortunately. In midguts, however, CYP9Q1 loci expression of Syriaca treatment group was significantly decreased compared to its controls, and no significant change was observed between control and treatment groups of subspecies in other cytochrome loci. When the treatment data that are normalized to control means were compared, again a very significant decrease in CYP9Q1 loci were determined in Syriaca compared to Caucasica, and again no significant change were observed in other loci. These 3 cytochrome P450 genes are reported to be specifically involved in comephos metabolism, and the fact that we found perizin treatment decreased CYP9Q1 activity in Syriaca indicate that Syriaca subspecies may have a less efficient metabolism of comephos, which makes it vulnerable to negative behavioral effects of perizin and also its active ingredient comephos. And the reason for the lack of a significant result in brain gene expression assays can be maybe sample sizes may not be adequate, they should be increased, or other neurotransmitter systems, not these ones, are involved in the behavioral effects of comephos. We think dopamine octopamine system can be important. It's an important neurotransmitter system in insects, and this system may be involved in the effects of perizin and comephos and should be further investigated. And that's all. Thanks for your patience.